It's the 8th of August, 2025. This is the Room Now podcast. Hi, I'm Dr. Jack Cush, executive editor of RoomNow.com. This week on the podcast, we'll be talking salaries, vaccines, drug repurposing, and how are you sleeping? I hope you're sleeping good. You know, that probably is one of the best vital signs I think we all have, we need to have. I found this study really interesting. It's about sleep. And what happens if you sleep badly 3 nights in a row? It's a meta-analysis of 35 studies, almost 900 people, and it shows that 3 consecutive nights of sleep deprivation defined as getting less than 4.3 hours sleep per night, was associated with a significant increase in IL-6 and C-reactive protein. I always say, sleep bad, feel bad. Bad sleep takes whatever you got wrong with you, just amplifies it, makes it all worse. I guess the question is it just the sleep's effects on the brain? Or is it got more pathology than that? Is there an element of neuroinflammation here? Anyway, a lot of studies proving the same point. A few reports this week on Scleroderma. One says that, and I saw this in Helio, and thank you, Helio, for putting this up there because I went and looked at the report. Greater than 30% of patients with systemic sclerosis present with no Raynaud's. This was based on 2 large cohort studies. One showed like 30%. The other one showed over 40% of patients who presented, were diagnosed with scleroderma, usually presenting with puffy fingers, musculoskeletal complaints, and then skin findings, but did not have Raynaud's at presentation. And what's different about those people? They tended to have more severe skin disease, and they tended to have more musculoskeletal complaints. You know, I don't like making the diagnosis without Raynaud's. I immediately go into my tight skin, other than scleroderma differential diagnosis. But this says it's certainly possible, and it's a sizable minority of people. The other report actually was just a review that said the good news is there's a big pipeline of drugs being developed for scleroderma. Most of us are not aware of this, but there are at least 9 companies that are developing fusion proteins, peptides, small molecules, monoclonal antibodies, polymers, gene therapy, and I'm not even including CAR T-cell therapies that are looking to include scleroderma patients in their trials. This comes from a lot of well-known and not-so-known companies: Bayer, Novartis, GSK, Boehringer Ingelheim, Celgene, J&J, Prometric, Cytori, Argentis. I like it. I want to see more research in scleroderma. Certainly, we need the help, do we not? Let's go on to another rarity, and that would be Cryoglobulinemic vasculitis. This retrospective study of 63 patients who either had essential mixed cryoglobulinemia with vasculitis or connective tissue disease-related cryoglobulinemia vasculitis showed that, number one, these people flare. You know, flares are part of the picture. So while in this study, they all went into remission with rituximab, and then they followed them out for a median of 58 months, and relapse rates at 1 year, 23%, 2 years, 42%, 5 years, 71%. Relapses were higher if they had purpura and had prior flares. These are difficult patients to manage. Rituximab obviously is a good induction therapy, but maintenance seems to be problematic. Most of these people were on maintenance therapy. A study this week compared vaccine responses on JAK inhibitors and those that were taking TNF inhibitors. So they're specifically looking at COVID vaccine, the SARS-CoV-2 vaccine, and this was not a humoral study where they're looking at, a 4-fold higher change in antibodies against the spike protein. This is a study looking at PBMCs and staining for gamma interferon production by CD4 T cells as a measure of vaccine responsiveness. And I think they had 22 patients on JAK inhibitors, 16 on TNF inhibitors. JAK inhibitor met criteria for a response in only 50%. Higher with TNF inhibitors, 81%. So, I have my rules. I tend to vaccinate everyone with two major, rules. And one is methotrexate, hold the methotrexate for a week. And the other one is rituximab or B-cell inhibition, vaccinate before or wait until 6 months after. The JAK inhibitors throughout COVID, patients that were on JAK inhibitors, I vaccinated them. I didn't stop. I think I probably would want to stop here. Again, if you did vaccine outcomes in multiple ways, you might get multiple results. But there have been hints that on a JAK inhibitor, you may not have the most optimal of vaccine responses. Last week we published a report on rheumatology salaries not being as high as you'd like them to be, that they're kind of flat. I added, that was data that was published on Medscape. I added into that data that was published by Becker's and published on Doximity. And then this week there's another report on your salaries. This one's coming from AMN Healthcare. AMN Healthcare did a long report on the demand on searches for certain kinds of specialties and what the salary is. Turns out the number one, internal medicine specialists or specialist MDs are in high demand in the United States. And that rheumatology is the seventh most in demand group. Yet when you look at the searches, we're like last in searches. Turns out that the 2024 salary low, there's a range here, is a high, a low, and a median. The low is $214,000. The high is $450,000. Congratulations, whoever you are. The median salary is $341,000. Who are these people? I don't seem to hang out with them or be employed by them. They also had a lot of information in there about signing bonuses, benefits, and whatnot. So if you're changing jobs, want to know what you're worth, I'd look at these reports, all three of them, Becker's, Medscape, and AMN Healthcare, and the link is in the notes. They also had a lot of information about advanced practice providers, NPs and PAs, where the average salary this last year, 2024, is $180,000. That's up almost 10% from $164,000 the year before. So while there's a big need for advanced practice providers, they're getting expensive, and I say they're worth it. This week there was a report on the use of patient-reported outcome measures, PROMs, a survey of almost 40 Spanish rheumatologists, 50 years of age, 10 years in practice. And while 80% of them agreed that there's an importance to doing PROMs in practice, only 28% were doing them routinely. And at that, they were mainly doing pain numbers, pain VASs, or a hack. The question is, why are we not doing them? And the number one reason that they've said, and that you said, is time. I do a lot of PROMs. I do factor them in. It's on my survey form. The patient fills it out while they're waiting to be seen. You should be using these. I like data about advances, especially in rheumatoid arthritis, potentially novel RA vaccine. And we've had RA vaccine studies. I did a vaccine study, 15 years ago that failed. Ranjini Thomas in Australia has been working on an RA vaccine with a lot of success. This one is an animal model where they developed a recombinant vaccine of HLA-DR beta 10401 bound to type 2 collagen, a type 2 collagen peptide that they modified chemically with a change in the glycine at position 265, and they showed that when they gave this vaccine to a collagen-induced animal model that expressed DR beta 10401, that they were able to suppress the development of arthritis. I like these results, except they're in mice, and I happen to treat humans. So people are working on vaccines. It's out there. It could be part of our future. In Japan, there's a famous large-scale IORRA study. This is an early entry of RA patients from 2007 to 2021. 10,000 patients have been enrolled. They looked at death. 915 deaths amongst the 10,000 that they followed for this 14-year period. That's about 10%. Deaths were from cancer, lung disease, cardiovascular disease, making up almost 60% of the deaths. Overall, these patients were protected as far as mortality outcomes if they were on methotrexate, if they were on a biologic or targeted synthetic, just a biologic DMARD, and it showed that low-dose glucocorticoids, again, makes mortality risk higher. So that's not good news. Another one on death here. Let me see if I can find this one. I don't know if I can find it. But anyway, next on my list is a meta-analysis. These are kind of oldies but goodies. These are points that have been made before that you probably know about, but I'm going to put them up there to remind you. The question is, do TNF inhibitors cause heart failure? Are you worried about heart failure with TNF inhibitors? It's a warning amongst the warnings that are out there. And that's based on largely Amgen and Centocor data, but there was a lot of data from Fred Wolf and other large cohort studies basically showing that RA patients with heart failure, that's well controlled, get TNF inhibitors all the time with no untoward effects. This large meta-analysis, 40 studies, 150,000 patients, TNF inhibitors being used in a multiple number of immune-mediated inflammatory diseases like RA, PSA, SPA, IBD, showed no increase in A, no de novo new onset heart failure, didn't happen, and B, people who had heart failure had no worsening when exposed to TNF inhibitors. Yeah, another oldie. I remember reviewing that data when I was on the FDA advisory panel back in the early 2000s. The same meeting, we actually reviewed the risk of significant LFT elevations in RA patients taking TNF inhibitors. It was a rare event, really rare. It was like among 6,000 people on infliximab, etanercept, and adalimumab, there was, I think, 160 reports. The bad news was that almost all of them were with infliximab. A few with adalimumab, but most of them with infliximab, most taking 5 to 10 milligrams a day, a lot of them complicated by background liver disease. So the question is, does this happen? So again, a small retrospective review of 9 patients taking etanercept 5, 4 on adalimumab. These were mostly female, BMI of 37, over 3 years follow-up. Their LFTs decline. Their fibrosis scores, the FIB4 scores, significantly decreased, and their albumin and bilirubin levels stayed stable. This is just a reminder that while you look at LFTs, I would say an RA patient on a TNF inhibitor with LFTs, I'd be looking at 9 other reasons why the LFTs are going the wrong way before I started considering the TNF inhibitor or stopping the TNF inhibitor. Another report that's kind of a oldie, but there's a lesson here, is lupus and its association with osteoporosis. This is a study of 110 osteoporosis patients. 110 lupus patients, and in that cohort, 40% had osteoporosis. In that cohort, 35% had lupus nephritis. So it turns out that lupus patients with low BMD and osteoporosis or osteopenia, that was associated with lupus nephritis class 3 and 4 disease, RNP antibodies, high CRP, longer disease duration, being older, abnormal HACs, and low complement levels. 83% were on steroids. That's they got lupus nephritis and they got osteoporosis and they got lupus, and it's all probably from steroids, is it not? But then again, let's think about it a different way. How many of your lupus patients, and the mean age in this cohort was 48, how many of your lupus patients, even those in their 40s, Are you thinking osteoporosis in the many things you do in managing your lupus patients? I don't, and I should. So it's not like I'm rushing out to do DEXA scans in my patients who are 31 and 28 with lupus and lupus nephritis. But if they've been on chronic steroids, maybe you should. This is a complication, and it's a bad one, with significant downstream effects. A report from Arthritis Research and Therapy looked at how often GCA patients have a normal CRP. Again, a head scratcher to me. A retrospective study from France, 380 patients with GCA, found that 29 of them, or 8%, had a normal baseline CRP of less than 10 milligrams per liter, less than 0.1 milligrams per deciliter at presentation, at baseline. GCA patients with a lower CRP level had less fever, more ocular complaints, especially anti-ischemic optic neuropathy. That was seen in 28% versus 13%. More limb claudication, 24% versus 8%. But it turns out high CRP or normal CRP patients had a same number of GCA relapses when followed out over time. So it does happen. It's got a slightly different presenting phenotype. Maybe the outcomes are the same. Again, these were all proven to have the diagnosis either by biopsy, CT scan, PET scan. Okay, most of those being biopsy. GM-CSF, there's been therapies that have been developed for GM-CSF as a treatment, anti-GM-CSF. It's a pro-inflammatory cytokine. Those therapies are kind of problematic and whatnot. This was a drug repurposing trial, and it looked at what drugs might have effects that would correct the GM-CSF excess. Turns out that CCL17, the chemokine CCL17, is a downstream mediator of GM-CSF, and in a drug repurposing exercise, they found 5 therapies that would have anti-CCL17 and therefore anti-GM-CSF effects, and these included, get your pen, paper, write these down, 2 of them you know of, fluoxetine, that's Prozac, ractopamine, Ractopamine, don't know it, ponesimod, don't know it, terbutaline, heard of that, and etravirine. They all are associated with inhibition of STAT5, IRF4, and CCL17, and in animal models, these drugs have been shown to have anti-inflammatory effects. I think drug repurposing is going to be a bigger thing in the future. The idea is there's been billions of drugs developed. A lot of them failed. A lot of them didn't work as well as you hoped that they would work. The question is, as we get smarter about genetics and AI and different ways of looking at how to use our older drugs better, smarter, drug repurposing is going to be an important drug discovery exercise. It mitigates the risk, the time, the cost associated with drug development. This is looking at molecular structures, looking at signatures, omics, lots of network-based analyses with machine learning. I think you'll be seeing more of this. I published a few months ago a drug repurposing exercise of drugs that were shown to probably have potential benefit in osteoarthritis, a disease for which we don't have effective therapies. I would look for these going forward, and I would expect machine learning to play an important role of that. My last report today is on the inefficiencies of ANA testing. This is an article from the Journal of Rheumatology this month. They looked at a single center cohort at a tertiary care center, and they researched the records of almost 2,500 individuals. And they found that in the ANA negative group, and only 271 or less than 10% of the 2,500 were ANA positive. In the ANA positive group, only 13% of the ANAs ordered led to a new diagnosis. Most of those were RA or spondyloarthritis. But it's a minority, a small minority. Like if you're considering spondyloarthritis or RA, why did you do the ANA? Obviously it's not you, it's other people. Again, these were ordered for many hundreds of reasons from many different sources, most being primary care and medicine subspecialties that are ordering these ANAs probably inappropriately. In those that were ANA negative group, only 1.6% led to a new diagnosis. That being, I got that wrong. In the ANA negative group, 1.6% led to a new diagnosis, most often RA and spondyloarthritis. In the ANA positive group, 14% led to a new diagnosis, them being Sjogren's, Lupus, and UCTD. Overall, 97% of these thousands of ANAs that were ordered did not lead to a rheumatic or autoimmune diagnosis. So again, this is stuff that's the kind of information you need to teach others about the appropriate use of the ANA going forward. I want to encourage you again to look at your inbox tomorrow, Saturday, over the weekend for the Rheum IQ. It's a cool little exercise. It takes a few minutes of your time. You get to see what you might have missed on Rheum Now this past week, what you should have learned. You don't always get them right. I'm surprised. I think these questions are pretty easy, but the performance characteristic shows a perfect bell-shaped curve with the average number of right answers around 3.5. And that's usually out of 6 or 7. One that a lot of you missed last week was a true-false. A South Korean study found that RA patients have a higher risk of gout. True or false? The answer is true. And I know, and you heard me talk about this last week on the podcast. I say you can't have both, but we know it does happen. In this particular study of 35,000 new RA patients compared to 103,000 controls showed that RA has a 2.7-fold higher risk of developing gout when followed over time. That's on Rheum IQ. You can take it next week and see how you compare to your peers. Take care of yourself. We'll talk next week.